Hello friends, I am Suchi. Welcome to my YouTube channel. In this video, we will discuss about Hershey-Chase experiment. Before that, you have to understand Griffith experiment and Avery-McLeod-McCarthy experiment. I have given the link of these two experiments in description box. Just go through it. In Griffith experiment, actually what happened? In 1928, while Frederick Griffith was working on Streptococcus pneumoniae, he found that some factor present in heat-killed strain of bacteria transforming non-virulent strain of bacteria into virulent form. But he didn't prove that DNA is the transforming factor present in a strain of bacteria which is heat killed and transforming this non-virulent strain of bacteria into virulent form. After him, Avery-McLeod-McCarthy were togetherly working on this transforming factor of Griffith experiment, and they concluded in their experiment that DNA is the transforming factor present in that heat killed strain of bacteria, converting this non-virulent strain of bacteria into virulent form, and it is the genetical material which is passing from one generation to next generation. But still many biologists are there, they believe protein is the genetical material which is passing from one generation to next generation, and it is the transforming factor in Griffith experiment. To know actually which one is the genetical material, DNA or protein, Alfred Hershey and Martha Chase in 1952 did one experiment by taking bacteriophages. What is this bacteriophages? This bacteriophages is a type of virus which infects and replicates its number within bacterial cell. So, bacteriophages is a type of virus which infect and replicates its number within bacterial cell. How? Suppose this one is a bacterial cell, it is having own genetical material. When virus come in contact with this bacterial cell, it will attach on the body surface of bacterial cell. Then what will happen? This virus will infect this bacterial cell and pass its genetical material into bacterial cell. So, what will happen? The genetical material of this virus will enter into bacterial cell. When replication process will occur within this bacterial cell will replicate this virus genetical material along with its own genetical material. So, within this bacterial cell, genetical material of bacterial cell will increase and virus genetical material also will increase. Then protein coat will form and genetical material will enter into this protein coat and complete virus will form within this bacterial cell. Then what will happen? When bacterial cell will get lyse, or when lysis process will occur, this bacterial cell will rupture, then complete this bacteriophages, or you can say virus, will release from bacterial body. This is the life cycle of bacteriophages. This is the transduction process. This is the transduction process. Then why Alfred Hershey and Martha Chase choose these bacteriophages for their experiment? Because this bacteriophages doesn't have own mechanism to replicate its own number. It need one host cell, which is bacterial cell. So for that reason they choose this bacteriophages for their experiment, and it's also having own DNA and protein coat. This outer coat is made up of protein. Both are present and they need both for their experiment, and it need also a host cell for replication. So for that reason they took these bacteriophages for their experiment. Now we will see how they did their experiment. For their experiment, Alfred Hershey and Martha Chase took bacteriophages and grew in two different nutrient medium. One nutrient medium contains radioactive phosphorus, another one nutrient medium contains radioactive sulfur. So in this case what they found? The bacteriophages grown in this medium absorb this radioactive phosphorus by their DNA. Why DNA absorb that radioactive phosphorus? Because DNA is made up of pentose sugar, nitrogenous base and phosphate group. Due to presence of this phosphate group, the DNA present in these bacteriophages absorb this radioactive phosphorus, but it didn't absorb this radioactive sulfur in this case, because the DNA do not contains sulfur. So in this case DNA absorb, but in this case DNA didn't absorb this radioactive sulfur. But in this case outer coat of bacteriophages, this part, didn't absorb this radioactive phosphorus. Why? Because outer coat of bacteriophages is made up of protein, and protein do not contains any phosphorus. It contains but sulfur. So in this case this outer coat absorb this radioactive sulfur, but here it didn't absorb this radioactive phosphorus. Get it clear? Once again I am repeating. In this case DNA absorb this radioactive phosphorus, but outer protein coat didn't absorb this radioactive phosphorus. But in this case DNA didn't absorb this radioactive sulfur, but protein outer coat absorb this radioactive sulfur. So what they found? They found radioactive phosphorus in DNA and radioactive sulfur in outer protein coat. They took two different group of bacteriophages. One contains radioactive phosphorus containing DNA, one group contains radioactive sulfur containing outer protein coat, and completed their experiment in three different steps. First step is infection, second step is blending, and third step is centrifugation. In first step they allow this radioactive phosphorus containing DNA bacteriophages to infect bacteria. Here they took bacteria, that one is E. coli. After infection, genetical matter present in this bacteriophages enter into E. coli cell. In this case also same thing happen. genetical matter enter into this E. coli cell. After infection, what they did? They did blending process. In blending process, what will happen? They separate this bacteriophages from the surface of E. coli body cell. So, they separate this bacteriophages from the surface of bacterial cell. During blending process, this bacteriophages got separate from the surface of bacterial cell. Then they did third step, that one is centrifugation process. What do you mean by centrifugation process? Centrifugation is a separation process in which molecule will get separate on the basis of their density. It means molecule which is heavier, it will be at the bottom of the vial, which is known as pellet, and molecule which is lighter, it is in supernatant. Supernatant, this is the liquid part or solution part. There you will find the lighter molecule. When they tested, they found that in pellet part, bacterial cell are there with bacteriophages genetical material. It means bacterial cell which is infected with genetical material of bacteriophages. It means in this pellet part you will find bacterial cell plus genetical material of bacteriophages. And here they found radioactive phosphorus. But in this case, when they tested this pellet part, they didn't found any radioactive content. No radioactive content. So, in their experiment, they concluded that DNA absorb this radioactive content, it get infected and introduced into the bacterial cell or enter into the bacterial cell. That's why in this pellet part it is showing this radioactive phosphorus, because here DNA is the genetical material, it is getting infected or it is entering into bacterial cell during infection and passing from one generation to next generation during transduction process. But such type of radioactive content they didn't find here, because here genetical material is getting transferred, but it is not having any radioactive content. Here radioactive content they found in this supernatant part, because in supernatant part this bacteriophages protein coat are there. So, they concluded that protein is not the genetical material, DNA is the genetical material which is getting transferred from one generation to next generation. This is all about Hershey-Chase experiment. Please like, share, comment and subscribe my channel.